Assalamu alaikum to all of you. I hope that all of you are quite well. Today my presentation topic is Silvestrol. My name is Raha Muslim and I am from the Department of BS Biochemistry. Today I am going to discuss about the discovery of Silvestrol, its structure, biological activities, ELF4 inhibitor, its mechanism of action, and the synthesis strategies of the Silvestrol, and the structural activity relationship to its analog, and the development efforts and the latest research. First of all, I am going to give you an overview on the discovery of the Silvestrol. So, the first rocagolate rocaglamide was reported in the less than 30 years ago, and this is a relatively recently discovered class of plant Algelia fovelata. This plant is mostly found in the rainforest of Southeast Asia and the countries like Malaysia, Indonesia. And the box of the plant Algelia fovelata contains silvestrol, which is a potential inhibitor of Ebola virus and Zika viruses. So what does the Ebola viruses and the Zika virus replication? Ebola virus, its virus causes severe inflammation and tissue damage throughout the body. Because it can cause problems with the clotting system of the body and lead to internal bleeding. Its outbreak occurred in West Africa in 2014. Zika virus replication takes place on the surface of endoplasmic reticulum in cytoplasmic viral factories. And the next one is the structure of the silvestrol. As I already told you, the silvestrol is a natural product from the family of Algelia fovelata, and it has a cyclobenzapurine and unusual dioxane ether side chains. These are the two structural diagrams, and with the help of these diagrams we can be able to see that how the dioxane ether side chains are attached with it. Next one is the function of the silvestrol. Silvestrol works as an potential inhibitor, as an anti-cancer virus agent, antiviral, and anti-inflammatory agent. So silvestrol was found to possess comparable cytotoxic activity against several human cancer cell lines to that of the well-known anti-cancer agents like the silvestrol. So what does mean of apoptosis and the L and CAPE cells? So apoptosis is the programmed cell death. And what does the term cancer means toward? If a cell apoptosis function is not working properly, the cell can grow and divide and ultimately create a tumor, and this is referred to be as a cancer. And Allen Cape cells are the cell lines in the human body, and it has a hydrogen receptor. Next one, what are the biological activities of the silvestrol? Silvestrol has very useful effect in the biological point of view as an anti-cancer, antiviral, and anti-inflammatory agent. So how does silvestrol works as an anti-cancer agent? Silvestrol is toxic against human cancer cell lines propagated in vitro or in vivo with a potency similar to that of the paclitaxel or camptothecin. It is a translation initiation inhibitor factor that prevents ribosomes loading on the messenger RNA template by targeting the eukaryotic initiation factor ELF4A. So, what does mean of in vitro and in vivo? In vitro is outside the living body or in an artificial environment. In vivo living organisms experiments, like the processes and the experiments that are performed on a living organism. For example, it can be a human being or an animal. So the next one is that The compound has been found to show promising in vitro and in vivo activities against certain B-cell malignancies. However, the mechanism of action of Silvestrol responsible for inducing cellular death is still unclear. The control of protein synthesis is essential for normal cellular function and survival, but unrestrained protein synthesis can promote tumorigenesis. Therefore, Silvestrol's ability to block protein synthesis is of significant interest in potentially treating cancers. In 2006, Swanson demonstrated that Silvestrol significantly induces alterations of 20 apoptosis and cell cycle related genes in Allen-Cab's cancer cells. It showed cytotoxic effects in Allen-Cab's cells by blocking the cell cycle is the G2-M control point. And G2 phase is a subphase of an interphase in which spindle formation occurs and cell grows and the cell prepares to go into the mitotic phase. And this is an important barrier for tumor formation in healthy cells as replication of cells with DNA damage is prevented. So what is ELF4A inhibitor? So the eukaryotic initiation factor 4A is an ATP-independent RNA helicase that is responsible for unwinding the secondary structure of messenger RNA. And in case of Silvestrol, the toxicity is reduced compared to its gold standard. Some virus replication relies on the host machinery so that they can be able to replicate its genetic material. And it is not surprising that Silvestrol and other flagellanes show antiviral activity. And this is the diagram, and this is the ribosome 5 to 3, and this is the coding region in which adenine, uracil, and guanine is attached. 40S is the smaller subunit of ribosome and 60X is the larger subunit of ribosome. And here is also the poly A binding proteins, and the Silvestrol is also attached. And this is also the diagram in which the messenger RNA is attached with the coding region and the LF4AF, and this is the recruitment blockage. So the next one is the antiviral agent. So as we already see that how it works as an anti-cancer agent, now we will see that how it works as an antiviral agent. Research studies has shown that it exhibits broad-spectrum antiviral activities against a variety of viruses, including the Ebola virus, Zika virus, dengue virus, and several types of coronavirus. The mechanism of action of Silvestrol involves targeting a protein called eukaryotic initiation factor, which is involved in the translation of proteins, viral proteins. By inhibiting this factor, Silvestrol disrupts the viral protein synthesis processes, thus inhibiting the viral replication. While Silvestrol has shown promising results in laboratory studies and animal models, it is important to know that it has not been approved for the human use and as an antiviral agent. The compound has several challenges associated with its development, including limited availability, low solubility, and potential toxicity concerns. Further research is needed to evaluate the safety and efficacy of Silvestrol in humans before it can be considered as a valuable antiviral treatment. So that how it works as an anti-inflammatory agent, the mechanism of action of Silvestrol involves its ability to inhibit a specific cellular process called as translation. Translation is the process by which messenger RNA molecules are translated into proteins. Silvestrol specifically targets a component of the translation machinery, which is called as the eukaryotic translation initiation factor 4A, which is involved in unwinding the messenger RNA secondary structure and facilitating the binding of ribosomes to the messenger RNA. By inhibiting this initiation factor, Silvestrol disrupts the translation process, leading to a reduction in the production of specific proteins that are necessary for the survival and growth of cancer cells or for the production of inflammatory mediators.